Welcome back, everyone, to the final talk of the day for day 2 of the FELF workshop. And it is my pleasure today to introduce the iGEM Évry Paris-Saclay team. For those of you who don't know the iGEM competition, it stands for the International Genetic Engineering Machine, and it's a competition that's organized annually. It was started in MIT many years ago, and it involves student teams carrying out a research project over the summer and then presenting this project at the iGEM Jamboree later this year. And today, the students from the iGEM Évry Paris-Saclay team, which I co-supervise, will present their project. And I will ask Maeva to take over from here.
Yes, hello everybody, I'm Maeva. So we are the Évry Paris-Saclay iGEM team. We are made up of 3 bachelor's students and 12 master's students. We are participating in the International Genetic Engineering Machine competition, in short, iGEM. And today, 5 of us will present you our project, which is on how to train your microbe for rosewood sensing. So there have been many new reports in the past few years that have raised the issue of rosewood trafficking. It is a major environmental issue of our times. The rosewood species are the most trafficked wildlife product in the world by volume and value. Since the ban of rosewoods, illegal logging 10 years ago in Madagascar, alone an estimated 2 million logs are still hidden in the northeastern part of the country, summing up to an equivalent of $300 million. In contrast to the noticeable elephant's ivory of tiger's furs on rosewood is logged, it is almost impossible to distinguish it from other non-protected wood species by the naked eyes, and still hard with the use of a microscope. However, there is compelling scientific evidence to believe that the Dalbergia species, the main rosewood species in Madagascar, can be distinguished at the genetic level with high precision. Yet, to the best of our knowledge, no rosewood detection tool based on genetic content exists. So, in order to tackle the problem of rosewood trafficking, we decided to build a rosewood biosensor that's based on DNA sequence.
Okay. So, hello everyone, I'm Daniel, and I'm at DNA script right now. So, how did we identify the unique rosewood signature that we'll be using for our sensor? So, as you see in the schematic, comparative genomic analysis can help reveal certain signatures in the DNA that are unique to a species. So for our first effort, we look for fully assembled genomes of the Dalbergia species in order to run comparative genomic analysis against other wood species. With the goal, of course, of finding certain genes that were either specific to the Dalbergia species or genes that showed enough differences to distinguish between Dalbergia and other wood species. So very much similar in the spirit of a barcode, as you see in the picture. However, we did not find full genomes of the Dalbergia species that were previously assembled. But we did find studies that had previously looked at the task of using phylogenetic analysis to accurately identify the Dalbergia species for conservation purposes. So from these published studies, we identified 3 genes that could serve as DNA signatures. That is, again, genes that provided sufficient genetic information for species identification, namely RBCL, MATK, and tRNAL. And with this, we built a data set that consisted of 300 samples from different Dalbergia species with sequences for these 3 genes. Okay? Now, currently, all Dalbergia species, and in particular those endemic to Madagascar, are protected by international regulations. So, with the aim of creating sensors that could detect the majority of illegal logwoods, we assembled consensus sequences for each of these genes that were representative, of course, of the Dalbergia species present in Madagascar. Okay, so at this point, we had the DNA signatures identified, and our goal was crystal clear. We wanted to design a DNA detection tool that could give us a fast and cheap output. So to achieve this goal, we decided to work with a molecular tool known as the toehold switch. But before going into details, explaining it further, I will zoom out a little bit to put the biology more into context. So the central dogma, the flow of information, as all of you know, DNA is the basic molecule of life that codes for all of the biological processes that happen inside each cell of an organism. And many of you might also be familiar with proteins, molecules consisting of amino acid residues that are the functional workhorses of the cell. But yet another fundamental molecule in life, in the process of life, is RNA. RNA also has very diverse functions, but for the purposes of this talk, let us just focus mainly on its role as an intermediate step between DNA and proteins. So what happens? DNA is transcribed into RNA by the RNA polymerase, and then, as beautifully shown to my right, the ribosome translate RNA into protein, and this process is called translation. Okay, so let us look a little bit more carefully into this last step process, translation. It turns out that RNAs also interact with each other through intramolecular and intermolecular forces. And these interactions give rise to different shapes, as you see in the figure, different shapes or different conformations of RNAs. And the key here is that the ribosomes will not be able to bind to some of these conformations because some of the bases of the RNA could be blocked geometrically. And we will use this fact, exactly. We will use this fact to cleverly design an RNA structure that can switch between conformations that bind and do not bind ribosomes in the presence of specific trigger sequences that, for our purposes, in our case, it will be exactly the previously identified DNA signatures. So let's get in more into what we did. Let's look at the toehold switch. Structurally, the toehold switch is simply an RNA hairpin structure that contains a region complementary to the trigger RNA on the left of the figure, a site of interaction where ribosomes will bind in the upper part of the figure, and then a downstream gene that will be expressed as a reporter in green to the right of the figure. In this native set, the toehold switch conformation blocks interactions with ribosomes. So in this native set, we see no output from the sensor. However, in the presence of trigger RNA, these molecules will bind to the toehold domain, again to the left, because they are complementary to them. And this will switch the conformation of the structure, opening it up, releasing the ribosome binding site in blue, or RBS, and consequently allowing the expression of the reporter gene, and of course, the output of the sensor. So just and I want to list some key properties of the toehold switches that make them excellent candidates for nucleic acid detection, so you may encounter them in the future. First, a high level of modulation in expression, in protein expression, has allowed us to achieve a very large dynamic range, over 2 orders of magnitude of dynamic range. Also, very importantly, they can be designed for very specific applications. So, for example, toehold switches have been designed for the diagnosis of various pathogenic viruses like Ebola, Zika, or norovirus, as written below. And for these purposes, the toehold switches have also been incorporated into cell-free paper-based platforms. And in these settings, researchers have achieved very fast detections, less than 2 hours, very low limits of detection, 30 nanomolar of trigger RNA, and also ultra high specificity, being able to distinguish of sequences with just 3 nucleotides of difference.
Okay, hello, my name is Tristan. I will tell you now how we build our total switches. As you already heard, RNA folds according to specific base pairing rules, adenine pairs with uracil and cytosine pairs with guanine. And to design RNA structures like total switches, softwares with RNA folding algorithms are used. And those algorithms are mostly based on thermodynamic models, which calculate base pairing probabilities. And those RNA sequence structure predictions can go in both directions. Firstly, the RNA folding, where an RNA sequence is known and a secondary structure is predicted. Secondly, inverse RNA folding, where a structure is known, often together with some sequence constraints, and a candidate sequence can be predicted. So we had two approaches of designing our switches. And our first approach was the usage of a web tool developed by an earlier participating iGEM team from Hong Kong. And the way it works is that it takes an input, our target Rosewood sequence, chops this sequence of various length into smaller fragments, and embeds those fragments each into a fixed switch sequence. Then it calls RNA fold, and the RNA fold algorithm, which takes a known sequence to predict an unknown structure. And it also gives us an output, the minimum free energy, like the thermodynamics of the folding of the trigger itself, the switch domains, and the trigger switch complex. And as an output, then you get various switches, which are then ranked according to an efficiency score calculated with a linear regression model, which itself is trained on switches from the literature, 181, where the so-called on-off rate, basically its metric is known to identify the most promising switch candidates. And the second approach for the design of our switches was the development of an in-house pipeline, which we call Total Designer. It uses a popular RNA structure prediction suite called NUPAC. And it works on the principle of inverse RNA folding, and it predicts toehold sequences with target Rosewood sequence and defined structures as an input. Those output switches are then ranked and chosen according to several matrices. This includes so-called translation initiation rates of the start codons of the stem hairpin of the total switch, and compares it to the report gene start codon. Then also takes into account calculated Hamming distances, which are the number of mismatches of our predicted structure to the desired structure, and in addition, minimum free energies of the various switch domains.
Hello, I'm Angeline. So, now that we have the tool to develop a sensor for DNA signature, we wanted to test it experimentally. And we decided to do that in the lab by introducing the genetic construct needed to test our system in bacterial cells. What you see on this figure are colonies of bacterial cells on the left that do not contain the trigger sequences, and those on the right that do. Clearly, fluorescence only appears when the presence of both switch and trigger, as in case B, is. Before testing our total switches on real Rosewood samples, we have to make sure that they show activation in presence of the connect trigger RNA. To do so, we read two different kinds of DNA plasmid constructs, one containing the short trigger sequences only, and the other the tall switch sequence followed by a GFP fluorescence reporter genes. Those are our full constructs, but how did we insert our DNA sequences into the plasmid? To insert our trigger and switch sequence into their corresponding plasmid, we use molecular cloning technique called Golden Gate. Here, the plasmid is cut using restriction enzymes at specific sites, and the sequence of insert is inserted by ligation into the backbone, producing the recombinant plasmid. In order to test the successful clone switch and triggers, we insert plasmid containing the switch together plasmid, the cognate trigger, into E. coli bacterial cells by co-transformation. Doing so, the trigger and the switch are transcribed simultaneously, and the switch is activated by the binding of the trigger. The activation can be detected through GFP fluorescence measurement. As a negative control, we can transform the switch expressive plasmid together with an empty plasmid that did not contain the cognate trigger. So this brings us back to our earlier slide. We obtain these colonies after co-transformation and look at them under blue light through a number filter. On the left is the negative control with the switch and an empty trigger. On the right, there are colonies with both of the switch and the trigger. Why such visual analysis is quick? We wanted to collect quantitative data about the difference in green fluorescence between the off and the on state. So, to get quantitative information about switch activation from cells, we group them in 96 well plates and measure their optical density of well as fluorescent intensity over many hours using a microplate reader. Fluorescence signal of switch trigger co-transformant was compared to the negative control. If the total switch worked as expected, the fluorescence signal of switch trigger co-transformant must be higher than that one of the negative control. This graph shows data collected by MATK switch 1.1. It shows the fluorescence intensity normalized through an optical density plot as a bar plot. We can see that without trigger, we have no fluorescent bacteria. We can see fluorescent bacteria with MATK switch 1.1 and trigger 1.1. We see similar data with other switch 1.3, where the switch activates only when the trigger is present. So, we can conclude that our MATK switches are functional. They work. This second graph shows data collected by switch LBSC 1.1 and 1.3. As with the previous set of switches, we can see that with no trigger, we have a little fluorescence, but we have much more fluorescence when bacteria also contain the cognate trigger. Again, our LBSC switches are functional. We also made one switch for TRML switch, and that one worked too. So, where do we go from here?
Thank you, Angeline. Hello, everyone. Here's Hassan. So after successfully testing the functionality of the in silico selected candidates in the lab, it's time now to translate this success from the lab to the field. And we want to apply this as an easy to use tool by the public authorities. And as you know, that living organism, they are intricate living organism, and they require continuous nurture in order to stay healthy and viable. So the final application should take into consideration that it should require the minimum technical expertise, if any. And needless to say, this biosensor should be, first of all, cheap and also gave rapid result to facilitate on-site use. And to meet all these requirements and to overcome the hurdles, a good way to do it is by implementing our sensor in free cell system. And as its name implies, cell-free system do not require any living cell, but only the bare essential components that carry out the gene expression. And briefly, as summarized here in this figure, so first of all, cells are grown, they are cultivated, and then lysed to extract to keep the cell extract and get rid of the genomic DNA in addition to the cell debris. And then another components indispensable for the genetic machinery, expression machinery, are added and mixed together will give us our cell-free system into which we can add our DNA for expression. So here, a result from our switches that are tested in cell-free system. Fluorescence is measured with respect to time. We have two cases where one, the switch was challenged with a trigger, and the second where the switch was only with water without a trigger. And as we expect that when the trigger is found, so we should notice or detect GFP signal as the green graph shows, whereas in the absence of the trigger, there's no GFP expression. And as you can see here, there's a quite remarkable difference between both signals that can allow us to confirm the presence of the nucleic acid in the sample tested. To wrap up all the story, we tested different switches. Can you move to thank you. So we tested here 6 switches. I call them just for simplicity A, B, C, D, E, and F. In addition to our negative control, the blue bar refers to the case where the switches were not challenged with a trigger, whereas the orange bar where the trigger was added to the switches. And if you can compare between those blue and orange bar in each case, you can find that they work well, except for E, which shows a constitutive expression and thus could not be used for our purpose. And the most promising one in our switches is the one called F, as you can see, as it represents a more than 4-fold increase or fold change between the one which is not activated and the one which is activated. And at last, to conclude, I can say that we tested the biosensors, and then we proved their functionality in vivo. First of all, we designed them in silico, and then we proved their functionality in vivo, and then in vitro using cell-free system. And the best switch showed around 4-fold increase in the presence of its cognate trigger or cognate signature that we selected in the very beginning. And we envision to translate this biosensor from the lab to the field by freeze drying this cell-free system that I just spoke about, which then can be easily handled and by the public authorities. So we can just use it on the paper disc or just keep them in the tubes so they can be easily handled and they don't need technical expertise. Just rehydrating with a sample with the water will allow this designed reaction to proceed and to yield the detectable signal. With that said, we reach the end of this presentation. I would like to thank you for your attention, and also many thanks for our supervisors, advisors, and for my colleagues and the iGEM team. With that said, questions, comments are welcomed.
Well, thank you very much. the floor is open to questions. I have a question from, well, I have a number of questions, in fact. Thomas, can you start?
Sure. First of all, thanks for the presentation. Really good. And we have a really cool application here. And then regarding this application, I have a question actually. So do you know what, at the moment, what the state of the art is that customs agent would use to detect, to know whether like some sample of wood is rosewood or not? Like, do they analyze the DNA, or what do they do? Do you know that?
Okay, I can give an answer about this. We met one professor from ETH who's working on rosewood, and he said that in order to really differentiate between rosewood and other trees such as ebony, it's very, very difficult. And if they want to do so, they go to the lab and they use the DART mass spectrometry to have this chemical signature, and then you can able to differentiate. But really, by the naked eye, it's quite like near from impossible.
Okay, awesome. Thank you.
Right. Okay. Thank you. Our next question comes from Corbin. Corbin, do you want to ask the question?
Sure. Thanks for the talk, guys. It was very nice. Maybe I'm missing something, but with the triggers, you're adding fragments of the Rosewood sequences, and with no trigger, you just don't add any sequence. Is that right? So I mean, I realize you have limited time to set up the project, so it could just be early steps, but have you considered using random sequences or sequences from other species as negative controls?
Yeah, I think Tristan can answer this question, but I can say yes, we challenged our trigger with different sequences other than the trigger that you used, but we did not show them. Tristan, can you give a brief answer about this?
Well, as far as I know, we also tested switches with, like we did cross-reactivity tests. We tested multiple switches having multiple triggered sequences and tested whether those switches also react to switches also react to triggers of that have a different complementary sequence. And depending on the switch, we got some so-called false positive activity, but depending on the switch, but the ones we showed, they showed good results.
Good, nice, thank you.
Our next question comes from Alex.
Yeah, thank you very much for the talk, and it's really cool work. I know that the iGEM teams have been really put under pressure because of all this, the pandemic, more pressure than normal. So well done. I was going to ask, have you considered using a readout other than fluorescence, as this something like a color change assay or a colorimetric readout of some form, because that would make it much easier to use in the field without having to carry some kind of excitation lamp and filters?
Yeah, as far as I know, those things were considered. Testing in plate reader colorimetric assays. The thing is, like the testing with the fluorescence readout using GFP as a reported gene has been tested before in our lab, and this is why, and quite successfully, and this is why it was like the closest approach, or we knew it can work quite well. And of course, in field, it makes sense to have it makes sense to think about having a colorimetric output, because it can, like, positive results could reveal better fold changes in the end than using GFP, and also it's more handy than even for cell-free in vitro systems to have a colorimetric output instead of a fluorescence readout.
Awesome, thank you. Well done, everyone.
Right, our next question is from Matthias.
Hello, thanks for the great talk. Thanks a lot. So I'm not sure I understood it correctly. So did I understand that the RIBO switches are binding to RNA and not DNA. Is this correct?
Yeah, right.
Okay, and if so, is there enough RNA there in the wood that is stable there, or do you transcribe first the DNA, or?
Well, thank you for this this good question. Actually, what we were doing in this project is just to, as a proof of principle for the feasibility of such kind of biosensor. But yeah, the main problem after this step is just to ask this question: are we able to extract enough DNA from the rosewood samples that are being trafficked, and then in order to challenge our switch? So this is the big question. Yeah, we have something in mind, but I think this is a question should be like kept after we can finalize, because the project is still ongoing in the lab for optimization. But yeah, this is the main challenge after finishing this step.
Okay, so it would be, for example, an option to transcribe in a cell-free the DNA to the RNA.
Yeah, exactly.
Okay.
So there's many, not many. I mean, there's techniques that are used in order to transcribe the DNA, such as NASPA or LAMP, that work also on room temperature. So this is not actually the problem. The problem is that are we able to have enough DNA that can be that can be compatible to the sensitivity of those techniques that can be used in order to amplify and transcribe them.
Thank you.
Thank you.
I would have a second question, if there's time.
Yeah, go on.
You showed a linear model in the very beginning to show the efficiency, and you said you trained it on some data. Can you elaborate on this? So I would be interested in which data you used, or which linear model you used, and so on.
This linear regression model was not done by us. It was done, maybe you could show the slides.
Yes, I'm on that slide, yes.
Thank you. Exactly, it was not done by us. It was done by the iGEM team of Hong Kong in 2017. And this linear regression model is trained. I have not gone deep into the details on how it was written, but basically it was trained on data sets from Collins' group from Harvard, which revealed a paper in 2014, publishing 181 toehold ribose switches and their scores. And based on those scores, or based on those switches, were analyzed, and metrics were calculated, metrics shown in this little window. And the regression model could basically calculate or predict weights, weights that show the importance of each of those metrics. And the way the scoring of the web tool works is that each of those predicted switches is analyzed in terms of that it's like minimal free energy values of the certain specific domains are calculated, and also some other parameters like the total domain paired basis, and those values are then weighted with those weighting values, and then this gives the efficiency score output, and then those switches are ranked according to this score.
Thank you. Thank you.
All right. Well, if there are no more questions, I think we will wrap up. Thank you all. Thank you all for being present. Thank you to the iGEM team for the wonderful work that you've done and the great presentation. Thanks a lot. We've had 2 great days of the workshop. We've listened to some great talks in synthetic biology, in distributed computing, and at the interface of these 2 fields. So it's been extremely exciting. This was the 2nd edition of the SELF workshop at DISC. So we are, well, we thank all the organizers of DISC for accommodating us and providing us all the logistical and technical sort of support to make this go smoothly. And we hope to continue doing the FELS workshops in the future. Please use the Zulip channels to give us your feedback. If you have any questions, if you want us to invite certain people in the future, do give us suggestions and we'll try to get the speakers here for the next year. Thank you all very much. Have a great weekend. Bye-bye.